Hi guys, my name is Rami, and I'm going to try to explain to you protein synthesis the best that I can. So, protein synthesis is basically the process of DNA turning into a protein. This process is also called the central dogma, which is DNA turning into RNA, turning into a protein. So there are two steps in this process. The first one is going to be transcription. And this process happens inside of the nucleus. Transcription is when the DNA turns into the RNA, and then the second step is going to be translation, where the RNA leaves the nucleus and turns into the actual protein. So the first step is going to be transcription, like I said. So the DNA is normally a double helix, like this. I tried to draw. So first, the cell partially unwinds the DNA where the gene is located so it can be read. And then special enzymes open the DNA, and RNA polymerase binds at the gene's promoter, which is the start signal. So, and then that goes into the second step, which is elongation. And in elongation, free ribonucleotides in the nucleoplasm pair with the exposed bases on the DNA template strand down here. And the RNA polymerase helps match them. So they have specific codes that match to each other. So T would bind to A to U, C to G, G to C, right? And then each added ribonucleotide is joined by a phosphator bond forming the RNA chain. RNA polymerase moves along the whole gene until the entire gene strand is copied. And then that would move into the third step, which is termination. Termination is when the RNA polymerase reaches the end of the gene and it would detach and the mRNA separates. And then the DNA is going to wind back up into its double helix. And so now we have the finished mRNA. It's a recipe copied from the DNA. So it's going to be the specific instructions to make a protein. And then that is leading into the second step of protein synthesis, which is translation. So the first step in translation is initiation. So in the cytosol, the small ribosomal subunit binds to the mRNA and scans to the start codon, AUG. So, and then a charged tRNA with anticodon UAC, which would match the AUG, joins into the P site. So this is going to come in and sit in the P site. And then the second step would be elongation, which is just going to be the repetition of this first process. So a charged tRNA whose anticodon is going to match the mRNA codon enters the A site next. Enters the A site. So another one is going to enter the A site right next to the P site. And its amino acid is going to form a bond with the amino acid on the P site. And so then this tRNA will move over into the E site, and it's gonna fall off. And the one on the A site is going to move into the P site, but it's going to keep that bond between that amino acid. And so it's just going to continue that process of scooting over and getting off until there's a long chain of amino acids all bonded together. So that leads into the third step, which is termination. So a stop codon is going to come into the A site, and a release factor will bond instead of a tRNA. And then the ribosomal subunits will separate, and then the new protein is released. And so then you'll have just your protein. So that's going to be the process. Maybe, I don't know. So that's going to be the process. And this process is really important to be like super specific because even just like one little mess up could cause a disease. An example of that would be a sickle cell anemia, the sickle cell anemia disease, and that would be a single base change in the DNA causes the wrong amino acid to be inserted during translation, which would lead to abnormally shaped red blood cells. So even just one change of a letter could cause a disease like having abnormally shaped red blood cells. So it's crucial for translation and transcription to be super accurate for producing functional proteins and just maintaining overall health. So, thank you.